You are most welcome, ladies and gentlemen, to the Understanding Spirit of Anatomy on Tuesday Night Anatomy with Daniel Okwan. I believe you are doing well. Today's section is going to be a quick revision, this time around looking at some aspects of gastrulation which takes place in the 3rd week of intrauterine life, together with some important cells. We call them, the 4th germ layer. Yes, the 4th germ layer. Okay, that is the neural Crest cells. We are going to look at that. Okay, so it's going to be a quick, revision. I have, some kind of 6 questions which in few minutes we are going to deal with them. And so, without much ado, let's set the ball rolling. Now, looking at question number 1, the first question says, before craniocaudal folding of the embryo, yes, just cranial to the buccal or to the oropharyngeal membrane is what structure? Okay, that is what we want to understand. I can just give you the answer now. The answer is going to be, of course, the cardiogenic area, that region, okay, where the heart is going to develop from. My friends, even cranial to the heart, we have what we call the undifferentiated portion of what we call the lateral plate mesoderm by way of what we call the septum transversum. So just cranial to the buccopharyngeal membrane or the oropharyngeal membrane, the future opening of the mouth is going to be, of course, this cardiogenic area. Now, just a quick, let me put some answers over here. Just a quick, reminder of ourselves. We got to know that the embryo around the 3rd week, this slipper shape, that is what we are aware of. Yes, so we realize that this is the cranial region or the head region where it's a bit broader, and then we have the tail or the caudal region. And then I told you that some proliferation of cells will take place from the caudal region going cranially. So let me see the proliferation taking place from here, from the caudal region going cranially. So that this time at the cranial region, we see this nodular mass formed over there. And one thing that we are aware also is that caudal, okay, over here we have the future opening of what we call the anal canal by way of the glaucal membrane. Glaucal membrane. Yes. And then this is the streak. Something we call primitive streak going through the groove, primitive streak. And one thing that you will be aware is that this primitive streak is the one which gives rise to all the 3 germ layers. The ectoderm, the mesoderm, the endoderm. Yes, be coming from this way. But to move from the caudal region and go cranially. So that the cranial region will form what we call primitive knots or the Hensen's node. Yes. And then emerging from the cranial region of the Hensen's node, we find what we call the notochord. We find what we call the notochord. Those are notochord. Okay, now just like we have glaucal membrane, the future opening of the mouth and the cranial region, that is what we call the buccal or the oropharyngeal membrane. One thing we are aware is that initially it will be what we call this oropharyngeal membrane initially was the procaudal plates. Yes, we've seen that procaudal plates. Okay, so that is what we are going to see. And I told you cranial to this, that is why I put this last sign over here. Like cranial to this, we are going to have what we call the cardiogenic area. Cardiogenic. Yes. So that's what we see. And one thing that we will also realize later is that even cranial to this developing heart will be the region we call undifferentiated lateral plate mesoderm, okay, which we've seen already. Something we call septum transversum. So the septum transversum will even be cranial okay, to it. Now, I'm talking about before folding, craniocaudal folding of the embryo. So that eventually with the folding, the heart eventually comes down, okay? This, central transverse is one of the derivatives, okay, of the diaphragm, for instance, so that the diaphragm will come below the heart. Eventually, that craniocaudal folding will ensure that these respective, structures get to their normal adult destinations or locations. Yeah, that's what we are going to see. Now let's look at number 2. Question number 2 says, what marks the beginning of gastrulation? My friends, one thing that we are aware about this gastrulation is that is the process which takes place in the 3rd week of intrauterine life, ensuring that we convert this primitive bilaminar embryonic disc, which is made up of 2 things, by way of the, of course, the epiblast and of course the hypoblast. Okay, these things will be converted so that at the end of the day we get the definitive germ layers. And whenever we talk about definitive germ layers, we are talking about the trilaminar embryonic disc. Okay, having the ectoderm, the mesoderm, the endoderm. Okay, that is what we see. So that is going to be, so the beginning, what is going to set the pace or going to mark this gastrulation process is, my friend, this guy we just mentioned. The proliferation of the cells, the epiblast cells, okay, this time around at the caudal region of the embryo and then moving cranially. That is what we call the primitive streak. So, my friends, when you are asked what marks the beginning of gastrulation, is the presence, okay, of the primitive streak marks the beginning of gastrulation. Yes, because at the end of the day, the first population of cells, okay, which will come, will ensure that they will displace the hypoblast, the primitive endoderm will be displaced, so that we form the definitive endoderm. The second population of cells, okay, that will come, will form what we call the mesoderm. And then the 3rd population will give us, will fill it, the ectoderm. Okay, that was what we are going to see. So the trilaminar embryonic disc formed from this primitive streak is formed in the succession, okay, beginning with, of course, endoderm, followed by mesoderm, and then ectoderm is the 3rd germ layer to be formed. Now, there's another question over here. It says, what is the effect of notochord on ectoderm? The effect of notochord on ectoderm. Yes. My friends, the answer is certainly right, because we've done all these things already. Is that the notochord will be making some, local, secretions or some paracrine product, okay, ensuring that they have effects on the ectoderm, converting the immediately overlying ectoderm to become what we call the neuroectoderm, so that we form the neural plates. Yes, that's the whole thing that I want you to know. Yes, so that is that one. Now, the next question is that what are the derivatives of the remnants of notochord? Now, notochord will also ensure that it forms the central longitudinal axis of chordates. Okay, now what is it going to do after it has formed? Because there is not going to be the final, central structure going to form the axis of the embryo. It's eventually going to be the vertebral column. So the point is, what will be the fate of this notochord? Okay, after it gets degenerated, the remnants, what will become of it? And last I told you about 3 things, okay, which I want you to know. That one of them, which is very important, I want you to know, is the nucleus pulposus of the intervertebral disc. Nucleus pulposus of intervertebral disc. Yes. So it means that they can give you the vertebral column and they will ask you, okay, what are certain things we can make up this vertebral column? Don't forget about the remnant of this notochord, because that remnant will give rise to nucleus pulposus of the intervertebral disc. Okay, that's one thing. The other one is that there's a ligament which projects from the dens, okay, or the odontoid process of C2, the axis, okay, cervical vertebra. And my friends, that thing is what we call apical ligament. Apical, so you've seen, the remnant by way of, we're talking about nucleus pulposus, apical ligament of dens. And then we have cranial, or the extension of this posterior longitudinal ligament over there. which we call it membrana tectoria. Okay, these three things. Okay, you know about them. But importantly, please know about the nucleus pulposus. The next one is that what is the primary embryological cause of something we call DiGeorge syndrome. Now, when we did this, I told you that one syndrome, which I made mention to you, was DiGeorge syndrome, where there's that kind of malformed phase. There's also issues with the products coming from, of course, the thyroid gland, and of course, coming from the parathyroid gland. Okay, that whole thing, including also some cardiovascular issues. Okay, some septum may fail to form. So what is the main embryological origin? Is the non-migration, the non-development, inability for neural crest cells to develop very well, and the ability to migrate to where they are supposed to be. That will lead to what we call this DiGeorge syndrome. That's the main embryological, problem that you have to be aware of. Yes. Now finally, the sixth question says, mention 10 derivatives of neural crest cells. Mention 10 derivatives of neural crest cells. Now let me just clean this. 10 derivatives of neural crest cells. Now already, I have told you that these neural crest cells, we can refer, because they give rise to a lot of structures, we now regard them as the fourth germ layer. Yes. So what are the derivatives? Now the last time I gave you some kind of mnemonic, okay, which will help you, which we said it is motel pass. Motel pass, okay. There are two S over here, there are more, but let me just put it this way. Now the M, I told you that there are a number of them. One of them I told you about melanocytes. Melanocytes. Yes, the pigment epithelium, except that of retinal pigment, will be coming from neural crest cells. I told you about these mesenteric nerve plexus together with messiness nerve plexus. Yes. Okay. That's why when you can find it in between the inner circular outer longitudinal muscle layers of the organs of the postpharyngeal gut. Okay, mesenteric. That's why it also has an eponym Orbes nerve plexus, and then messiness nerve plexus, which is also called submucosal nerve plexus, can be found over there. Yes, we talked about odontoblasts, okay, or the dentine cells, okay, dentine cells. Cell that will form the dentine. I told you about the T to be the tracheal cartilages, all these C-shaped rings of cartilages in there, okay, we talked about them. And I told you that the E, we don't need to worry about it. Now, what is the reason? Although some people write enteroendocrine, I am interested in, enterochromaffin cells. No, it's not enteroendochromaffin cells. I am interested in chromaffin cells, or the chromaffin-like cells, which we find in the adrenal medulla. Yes, that's what I'm interested in, the adrenal medulla. Now, I also told you about the laryngeal cartilages. Laryngeal cartilages. Okay. Then, we also talk about, now, the T over here, I want to add another thing to it. The T over here. Let's add this to it, which I didn't give you the last time. Now, just know about the tunica media. Tunica media, this time around. Tunica media of outflowing the proximal portions of the outflowing vessels of the heart. And when I want to talk about the outflowing vessels of the heart, I am interested in talking about the pulmonary trunk, Talking about the ascending aorta. Yes. Now the P, we can put a number of them over here. We talk about the parafollicular C cells. Yes. We talk about the pia mater. We talk about the arachnoid. Now together, so for instance, in an FME may be asked, leptomeninges are derived from what? Leptomeninges, because these two will become what we call leptomeninges. We call the leptomeninges. Yes. That is what we are going to see. Yes. Now, how about the A? A, we've seen arachnoid matter, okay, to be part of it, part of the leptomeninges. We have adrenal medulla, which I told you from the X-ray nuclear cells, adrenal medulla. From the, chromatophyll cells, adrenal medulla. Now, is that all? No. We're talking about all ganglia. Yes. Now, that's a big statement. All ganglia. Because if these cells are in the central nervous system, except of course the basal ganglia in the brain, then of course there will be, of course, from the peripheral nervous system, collection of cell bodies in the PNS, ganglia. So for instance, anything PNS, peripheral nervous, think about neural crest cells. Okay, so we can talk about all ganglia. Dorsal root ganglia. Talk about these, parasympathetic ganglia, ciliary ganglia, optic ganglia, okay, submandibular ganglia, okay, pterygoparietal ganglia. All these ganglia, they will be coming from here. I told you about some, sensory ganglia. Okay, I told you about the fifth, that of the fifth, the seventh, the ninth, the 10th cranial nerves. They are all coming here. Then we have the S. As last time I told you about thinking about the Schwann cells doing the myelination for axons of the peripheral nervous system. Schwann cells included the cell line cells. Yes. That's what we said. Then the S. There's another S, which I told you very important, septum. Septum. Now, not just any ordinary septum, but I'm talking about conotruncal. Conotruncal septum. Conotruncal septum. Yes, conotruncal septum. Why? Because we have the conus of the heart, and over there there's a septum who is going to separate the conus of the heart. And that septum is known as bulbar septum. Bulbar. B-U-L-B-A-R. Bulbar septum. And then that's truncus. Now we have the truncus arteriosus, the developing heart tube. We have the most cranial region, we call it truncus arteriosus. That portion will have to divide so that we get ascending aorta and pulmonary trunk. Now, that truncal part, okay, the septum over there will therefore be called aortico-pulmonary septum. Aorta and pulmonary trunk, so aortico-pulmonary septum. And that septum is spirals in its course. That is why we call it spiral septum. So you have conotruncal septum, you may see bulbar septum, and then you also see spiral septum. Together will form this conotruncal septum. Yes, that is what we are going to see. And then there's one more thing I want to tell you. Even the choroid and sclera of the eye. Choroid and sclera. So another S. So you can talk about the sclera and choroid. Sclera and choroid. They belong towards the eye. Yes, will be derived from, of course, these neural crest cells. I told about the nerves of the pharyngeal arches will be derived from neural crest cells. Yes. Okay. These are the things that I want you to know. So my friends, I'm very grateful for your time this evening. I hope you find it helpful. Yes, next week we'll look at another revision exercise for the same general embryology so that you can be well, prepared for whatever is up. Very grateful for your time this evening. Have a good night, all of you, and may the good Lord richly bless you. Amen.